Good morning, students. Last class, I discussed the chapter Microbes in Human Welfare. Microbes in Human Welfare. In that chapter, I will discuss clearly about useful microorganisms, microbes in human welfare. In that chapter, I will discuss clearly about useful microorganisms to mankind. So various microorganisms like bacteria, fungi and protozoans are benefit to human mankind, benefit to human beings, benefit to human beings. So these microorganisms, various types of microorganisms like bacteria and protozoans and fungi are used in the production of household products. By using microbes, we produced various household products like bread, dosa, idli, so and so on. And the yeast, so generally, naturally occurring yeast, so Saccharomyces cerevisiae is used in the manufacture of bread in the bakery. And the yeast is also used in the preparation of beer, whiskey and rum in the beverages, in the alcoholic beverages. And these microorganisms are also used in the manufacture of different type of cheese, Roquefort cheese, Swiss cheese. These are the important household products are produced by using microorganisms. And so microorganisms like eubacteria and fungi and actinomycetes are used for the preparation of various types of antibiotics. Various types of antibiotics are produced by using the important microorganisms like eubacteria, actinomycetes and fungi. So, by these microorganisms, various antibiotics, for example, bacitracin, polymyxin B, gramicidin, actinomycin, chloromycin, terramycin, neomycin, penicillin, clavicepsis, like various antibiotics are manufactured by using the microorganisms. And these microorganisms are also used. These microorganisms are also used in the manufacture of various organic acids, bioactive molecules and enzymes. The organic acids like citric acid, acetic acid, lactic acid, butyric acid, these are the organic acids. These acids are prepared or manufactured by using a different type of microbes in the industries for the large scale production. Then some enzymes, important enzymes like lipases, pectinases, proteases. These are the enzymes. Streptokinase is also an enzyme. These enzymes are produced in the industries by using different kinds of microorganisms. And bioactive molecules, some important bioactive molecules like statins and cyclosporin A and statins, these two are important bioactive molecules. They are also produced by using microorganisms. Several microorganisms are also used in the biofertilizers. Some microorganisms play very important role or employed very important role in the preparation of biofertilizers. So means these microorganisms enhances. These microorganisms are used in the manufacture of biofertilizers. These biofertilizers are enhances the nutrient quality of the soil. Means the biofertilizers contains microbes. The biofertilizers contains microbes or microbes present or microbes used in the manufacture of fertilizers. These fertilizers are called biofertilizers. The biofertilizers increases quality of the soil. Increases the quality of the soil. Quality of the soil. Or increases or enhances the quality of the soil. For example, some bacteria, free-living bacteria like Azotobacter, Azospirillum can successfully fix the atmospheric nitrogen. Then some rhizobium is present in the roots of higher plants. It also plays very important role in fixing the atmospheric nitrogen. And Anomina, Azole, Nasta, Oscillataria, Arosira. These are all the cyanobacteria. They can fix the atmospheric nitrogen. Clear? So then some microbes are also used as a biocontrol agents. Some microbes are also used as a biocontrol agents. For example, lady beetle is used. Lady beetle is used in the control of aphids. Dragonflies are used in control of mosquito, and spores of Thuringiensis is used to kill the butterfly larvae. And Trichoderma is used to control the soil-borne diseases like stem rot, root rot, and rhizome. And baculoviruses like nucleopolyhedroviruses are used to control the insects and arthropods. So, very interestingly, last concept, the microbes are also used in the sewage treatment. So, last class, all these concepts are discussed here in the chapter microbes in human welfare. Clear? So now I will start another topic or another new chapter, principles of inheritance and variation. Simply genetics. Principle of inheritance and variation. Simply it's called genetics. So what is genetics? So I will start with a new chapter, the principle of inheritance and variation. So simply this chapter is called genetics. What is genetics? Genetics is a branch of biology. Genetics is a branch of biology which deals with systematic study of inheritance. Systematic study of inheritance and variation. Inheritance and variation is called genetics. Here, the branch of biology which deals with systematic study of inheritance. Systematic study of inheritance and variation. Is called genetics. Clear? The branch of biology which deals with systematic study of inheritance and variation. That term genetics was coined by Bates. The term genetics was coined by Bateson, B-A-T-E-S-O-N, Bateson. Here, the term genetics was coined by Bayes. Genetics deals with two aspects. Genetics deals with two aspects. First aspect is inheritance and the second aspect is variation. Inheritance. What is inheritance? What is inheritance? Inheritance is a process. Inheritance is a process by which the parental characters can pass to their offspring. Means the parental characters are passed to their offspring is called inheritance. Clear? Inheritance is the process by which the parental characters passed to their offspring, is called inheritance. So we know that it's a general common phenomenon in all the living organisms. Common or general phenomenon or general process it occurs in all living organisms. Means the offspring resembles their parents. That is it. The offspring resemble their parents. For example, the height of the parent, color of the parent, structure of the egg. These characters, behavior also. These characters from the parents passed to their offspring from generation to generation is called inheritance. Inheritance means the offspring resembles their parents because offspring resembles their parents because the parental characters are passed to their offspring. This is called inheritance. Next, what is variations? What is variation? Variations means differences. Variation means differences. Here, variation means differences. Means the offspring not exactly alike to their parents. Means not exactly alike. The offspring. So first point I will discuss inheritance. Means parental character are passed to their offspring. Generally, the offspring resemble their parents, but genetics also deals with variations. Genetics also deals with variations. Variations means differences. Variation means differences. So the offspring not exactly alike. Got the point? The offspring not exactly alike to their parents, but the offspring shows differences. The offspring shows differences to their parents. It is called variations. The offspring are not alike. The offspring are not alike to their parents, but the offspring shows the differences. Offspring shows the differences. These are called variation. It's called variation. Next, what is heredity? Heredity. So, the heredity means the offspring resemble their parents. The offspring resemble their parents. It is called heredity. The offsprings resemble their parents is called heredity. Clear? Very important concept in this chapter, principles of inheritance and variation. So first one, inheritance and variation. What is inheritance? Just inheritance is parental character transfer to their offsprings. Then variation, differences. Offsprings, not all offsprings are not alike to their parents. Next, the term genetics was coined by Bates. Then the heredity, the offsprings are resembled to their parents. Offsprings are resembled to their parents. It is called heredity. So inheritance, variation, Bateson and heredity. Clear? So next concept, Mendel's principles. Next concept, very first, who work very first in the field of genetics. It's very important and very interesting. Very first, who started the work in the field of genetics? Who work in the field of genetics? We know the Austrian monk. The Austrian Gregor John Mendel. Gregor John Mendel. Gregor John Mendel. Start the work in the field of genetics. In the field of genetics. Clear? Very first, the Austrian monk started the work in the field of genetics. And Gregor John Mendel conducted hybridization experiment. Mendel conducted hybridization experiment on Pisum sativum pea plants nearly about 7 years. Mendel conducted hybridization experiments. Hybridization experiments on garden plant called Pisum sativum. Pea plant. Pisum sativum pea plant. Clear? Mendel conducted hybridization experiment on Pisum sativum pea plants about 7 years. About 7 years. So, from 1856 to 1863, in 7 years. 1856 to 1863 means 7 years he conducted the experiment on garden pea plants, nothing but Pisum sativum. So 7 years. Then Gregor John Mendel was born in 1882. Mendel was born in 1882 in Moravia. He was born in Moravia in 1882. And Gregor John Mendel was considered as father of modern genetics. He was designated as Gregor Mendel was designated as father of modern genetics. Father of modern genetics. Father of modern genetics. Father of modern genetics. Here, Mendel was designated as father of modern genetics. And Mendel on 8th February 1865. Mendel is 8th February 1865. Presented his results on Pisum sativa. 2. Presented his results on Pisum sativa. 2. Society for the study of natural science in Broome. BRUNN in Broome. His results conducted on or obtained by hybridization experiments conducted on Pisum sativa is published in 8th February 1865 to Society for the Study of Natural Science of Life. But at this time, the Mendel work was unnoticed. By the scientific world. And Mendel died. So his work was not so identified by the scientific world, and Mendel was died. And after death, he was regarded as the father of modern genetics. After death, he was regarded or honored as father of modern genetics. And he was died. So Mendel was died in 1884. Mendel was died in 1884. And after death of Mendel, he was regarded or honored as father of modern genetics. Yeah. So then, after the death of Mendel, the team of three scientists like Hugo de Vries, Karl Poinz, and Teschmark rediscovered the Mendel principles in 1900. So, in 1900, Mendel's principles of inheritance was rediscovered by the team of three scientists like Hugo de Vries, Karl Poinz, and the last one, Teschmark. These three scientists rediscovered in 1900 all the principles of Mendel, and all the principles are correct. That's why after the death of Mendel, he was designated as the father of modern genetics. Clear? In the 8th February 1865, it is very important. He presented his results on hybridization experiment conducted on Pisum sativa to Society for the Study of Natural Science in Broome. But at this time, his work was not unnoticed. For that time, Mendel work was unnoticed by scientific world, and Mendel was died in 1884. After the death of Mendel, three team scientists in 2019, Hugo Dewas, Carl Owens and Deschamps rediscovered all the Mendelian principles, and all the Mendelian principles are correct. That's why after the death, Mendel was regarded or designated as a father of modern genetics. Clear? Then Mendel, why Mendel selected pea plants for his experiment? This is very important, interesting question. Why Mendel selected pea plants for his experiments? Why Mendel selected the pea plants for his experiments? So, because of the following reasons, the Mendel selected the pea plants for his experiments. Take one by one. Mendel selected Mendel selected the pea plants for his experiments. Pea plants are easily cultivated. Pea plants are easily cultivated and does not require after K. This is the first point. Clear? So pea plants are easily cultivated and not required after K. So pea plants are annual and give results within a year. Pea plants are annual and give results within a year. Okay? And pea plants are bisexual. Pea plants are bisexual and cell pollination takes place. Cell pollination takes place. And in pea plants, the cross pollination is made manually. Suppose you want to cross pollination in pea plants, we can make it in pea plants manually. And in pea plants, several true or pure line breeding varieties are observed. So last one, the pea plants are fertile. The hybrid pea plants are fertile and produces the seeds, fertile seeds. Because of these reasons, Mendel selected pea plants for his experiments. First point is Mendel selected the pea plants because of they are easily cultivated. Pea plants are easily cultivated. And they do not require after K. Not required after K. Here. So easily cultivated. So then easily cultivated and not required after K. Second, pea plants are annual. Pea plants are annual and give results within the year. the year. Give results within the year. And pea plants are bisexual. Pea plants are bisexual. And cell pollination is carried out in the pea plants. Cell pollination occurs because pea plants are bisexual. And cross pollination is made by manually. Cross pollination is made by manually. Cross pollination is made manually. And the hybrid plants are fertile. The hybrid plants are fertile. Hybrid plants are fertile and produce the fertile seeds. Hybrid plants are produced. The hybrid plants are produced the fertile seeds. Hybrid plants are produced fertile seeds. And last point, the true or pure line breeding varieties. True or pure line breeding varieties have various contrasting characters. Various various contrasting characters are true or pure line breeding variety. Various observable contrasting characters are available in the pea plants. By all these reasons, Mendel selected the pea plants for his experiments. Then, Mendel selected the 7 pair of contrasting characters for his experiments. Finally, so Mendel selected 7 pair of contrasting character for his experiment. Mendel selected 7 pair of contrasting character for his experiment. They are taken characters and contrasting character So, in contrasting character, dominant and recessive characters. Dominant and recessive character. First one, stem height. First one is stem height. So, dominant is top and dwarf is recessive. Mendel totally selected 7 pair of contrasting character for his experiment. First one, stem height, tall and dwarf. Flower position. Axial and terminal. And third one is flower color, rubber color, white and white and white. Next, fourth one, pod means fruit color, pod or fruit color. So green and yellow, green and yellow. Next, fifth one, pod shape, pod or shape, inflated and constricted Then sixth one, seed color, yellow and green, So last seven pair of contrasting character, seed shape, round and ring, round and ring. Here, these are the seven pair of contrasting character selected by Mendel for his experiment. Stem height, tall and dwarf. Flower position, axial is dominant, terminal is recessive. Flower color, violet dominant, white is recessive. Then pod, fruit color, so green is dominant, yellow is recessive. Pod shape, inflated is dominant, constricted is recessive. And seed color, yellow dominant, green recessive. And seed shape, rounded, green cut. Here, these are the seven pair of contrasting characters, so selected by the Mendel for his experiment. Okay, thank you. Next class, I will discuss terminology, some important terms used in genetics. So some important. These are the seven pair of contrasting characters. Some important. So seven pair of contrasting characters selected by the Mendel for his experiment. These are all dominant characters. These are all recessive characters. Clear? So next, some important terminologies used in the genetics. Some important terms, terminologies used in genetics. Terminology. The important terms used in the genetics. So first one, the first term is true or pure line breeding variety. The first term used in genetics is true or pure line breeding variety. True or pure line breeding variety. So, what is true or pure line breeding variety? The variety in which the offsprings have the variety in which the offsprings have same traits Same types of parents. Same types of parents. On continuous self-pollination. On continuous self-pollination. On continuous self-pollination. On continuous self-pollination is called pure line breeding variety. Or true variety. The variety of offsprings, the variety in which the offsprings have traits means characters of parents. They have traits of parents. That means the offsprings have their parental characters. Very simple. The characters can pass from parents to the offsprings. That's why generally the offsprings or siblings can have the traits of parents, or they resemble to their parents, because the characters are passed from parents to the offsprings. On continuous self-pollination, this condition on continuous self-pollination, the variety in which the offsprings have traits of parents on continuous self-pollination. For example, the tall pea plants on continuous self-pollination. Produces tall pea plants. Tall pea plants are produced in all generations. The tall pea plants are produced. It's called true or pure line breeding variety. Clear? So second one, contrasting character. Contrasting character. The second one is contrasting characters. Contrasting characters means an alternative expression of the same character. Alternative expression of the same character. Alternative expression of the same character. Alternative expression of the same character is called contrasting characters. Okay? Contrasting means alternative expression of the same character. For example, the tallness of the pea plants is controlled by two contrasting genes, two pairs of contrasting genes. For example, T for tallness, and small t for dwarfness. So for example, tallness is controlled by two forms of alternative genes. For example, capital T, tallness, and small t, dwarfness. Here, this is about contrasting characters. So, okay, thank you. Next class, I will continue the remaining terminologies used in